On problem number 15 here we have an actual carbon-13 spectrum given to us. The main thing I've indicated here is that we have 3 signals present there. One at 20 ppm and then a couple above 120 here. Not important. Most important thing here is that we have 3 unique signals, and we're going to use that to figure out which isomer we have of these molecules here, this molecular formula. So we have 1, 2, 3. This one we did in the previous problem here. 3 signals with this one. You can see that's the answer because we have 3 signals given. I should say this is 3 expected signals, and that is what's given over here, unless we have overlapping peaks. But these others, you can see I've labeled them 5 signals for that, 4 signals for that one, and 6 for this one. These are going to be quite a few signals for those. And here we do expect that all the signals should really be apparent with this small molecule. So we expect this is the best answer for this carbon-13, just the number of signals for that. Okay. On problem number 16 here, this is asking for a chromophore. A chromophore is usually a pi bond or a lone pair that will absorb the UV visible light. And here this one is saying that which compound would absorb in the colored region. You should also know that the colored region is about 400 to 800 nanometers in its wavelength. So here this one in the colored region is something that contains lots of chromophores, and specifically conjugated double bonds. That's what this colored region is telling us. The more conjugated double bonds we have, in the case of D here, then the higher the wavelength that's going to be absorbed. And that was a principle that we learned. The more conjugated double bonds, the higher the wavelength absorb, or the lower the energy that is absorbed because of the conjugation. So you see here D has lots of conjugated bonds. That is a azulene compound. That is a actual blue compound from various organisms, a mushroom. Over here we have no conjugated double bond. The bond right here is expected to absorb in the UV region only, around 280. We do have an aromatic compound here, but we don't have as many of the conjugated pi bonds there, chromophores. And we also have just 2 over here. So if we select the best answer, it would be the one that would absorb the most here, at the highest number, I should say. Okay, next one here. Proton NMR. With Proton NMR, we're not selecting the carbon atoms that do not have a proton. So you can see I've labeled those 1, 2, 3, 4. Symmetry on this side of the ring, so we don't count it. That one's equivalent to that one, 2 is equivalent to this side, and 3 is equivalent to this side. Okay, a little bit faster. I got 1 through 3, and then 4, 5, 6. I should point out, don't get tricked by these right here. There's only one possible connection there. You can see to the oxygen. There's no hydrogen present. This one here is kind of tricky. We got a methyl group there. There is a hydrogen right here, don't forget about it. And then the 3 and 4 are equivalent to this side of the ring, and then there's another hydrogen right here on the oxygen. So that makes 5 altogether. This one has lots of symmetry down the middle of this here. And then you can see 1 and 2 for that.